Hi, my name is Tenny.
And my name is Manelli, and we're going to talk about gene flow and FST.
5 causes that change the allele frequency are natural selection, non-random mating, mutations, small sample size, and gene flow, which we're going to focus on. So, what is gene flow? Gene flow, also known as migration, is the movement of alleles from one population to another, which occurs, for example, when individuals, pollens, or seeds leave one population and join another, and they reproduce in that new population.
If genes are carried to a population where those genes previously did not exist, it could cause genetic variation. For example, in the picture below, migration of western deer to eastern deer population introduces a new allele to the eastern population.
Gene flow is not perfect, and there are restrictions to gene flow. Physical barriers to gene flow are usually but not always natural. They may include impassable mountain ranges, oceans, or vast deserts. However, in some cases, they can be artificial or man-made barriers, such as the Great Wall of China. For our example purposes, imagine a population in the oceanic water in which there are sexual reproduction and variations. However, when the oceanic water levels dropped enough and the Isthmus of Panama became a complete piece of land, it separated the eastern Pacific Oceans from the Caribbean Sea. This caused what used to be a single interbreeding population to become into two separate populations without gene flow. Another aspect of the gene flow is that if the rate of the flow actually affects the gene pool, and the answer is that yes, the direction and the rate of the flow actually affects the gene pool. For example, if the movement of allele is from population A to B, what happens is that A is not changing, but B is changing because we're introducing new alleles to the population B. So if the amount of allele exchange is the same both ways, then both populations are becoming more similar to each other, and that population, in other words, it becomes homogeneous.
Now we're going to talk about FST. FST is a statistic that we use to measure the variation among subpopulations. It allows us to know how distinct or similar two subpopulations are with each other. FST has a range between 0 and 1. In future slides, we will explain what each range indicates. First, we need to know how to calculate the FST value. In general, FST equals the difference of variation between populations from the variation within populations over the variation within population. This is looking at the proportion of genetic variation that it is within that subpopulation relative to that which is between that subpopulations. FST is measured based on heterozygosity, which is a measure of genetic variation. Heterozygosity is simply calculated from 1 minus sum of allele frequency squared. Now we're going to compare the FST ranges. In this example, the FST equals 1. This indicates that there is no overlap at all in the allele frequency of the subpopulations. In this example, in population 1, all the fish have the allele A, and in population 2, all the fish have the allele B. Thus, they are very distinct populations. In this case, where FST value equals 0, there is complete similarity between two subpopulations. This means that there is exact number of A alleles and exact number of B alleles in two subpopulations. In another case where FST has a value between 0 and 1 is an example of an intermediate case. The value of FST indicates the amount of differentiation within the subpopulations. Therefore, to conclude, high FST values indicate distinct subpopulations, where low FST values indicate homogeneous subpopulations.